Okay, welcome everybody. I'm so glad you guys were able to be here on this beautiful July morning. Those of you who are online, hopefully you guys can hear me all okay. So, my name is Amanda Rameriz. I'm actually from Tulsa. I teach for Union Public Schools, and today I'm going to be talking about microplastic particles into the agricultural soils through the application of biosolids. So going into that. Okay, so what exactly are microplastics? So microplastics are tiny particles of plastic that are smaller than 1 millimeter in size. They can vary in size, shape, and morphology, everything from fibers to pellets to even different films. Thank you very much. Okay, so then what are biosolids? So biosolids are semi-solid, nutrient-rich organic materials resulting from domestic sewage treatments from wastewater treatment facilities. About 23.3 billion kilograms of biosolids are produced each year in the United States. 95 to 99% of microplastics from municipal wastewater are captured in those biosolids. Okay, so as those biosolids are being generated, so how are they got to go somewhere. So what they do is, since they are so nutrient rich, they decided that, well, it sounds good to add to agricultural fields. As biosolids are separated from the wastewater, like I said, they got to go somewhere, and 53%, or about half of that 23 billion of all municipal solids produced, are actually being added to land throughout the United States. But so you got to think about that. So how many microplastics really are we putting out there? As we looked at some different literature, we saw some different number of microplastic solids from different studies that have been done. There is about an average of 469.5 particles between all of these different studies. Okay. This is ending up being a, I apologize. There we go. All right. As people have studied this throughout, the literature ranges over a large magnitude of differences. What we did this summer is, when we worked in the lab with Dr. Nanny in collaboration with the USGS, we developed a comprehensive and robust method that looks at the entire sample, and we found that a conservative number of about 4,800 particles per gram of dry weight, which is a lot more than what a bunch of other people have been able to find. Okay, so from this we were able to look at a quantification of microplastic particles using a digital fluorescent microscopy. We applied this through a bunch of different samples, and we looked at some light fractions, heavy solid fractions, and residual. And we compared this to the actual method of the NOAA, which is what as a lot of others have been doing. I don't know if you can see it because there's a thing in the way, but they were able to find, from using the NOAA method, we found about 65 particles. But when we used this modified method from that Dr. Nanny was able to comprise, all right, so then what is actually happening to microplastics after the biosolid application to the agricultural, non-food agricultural fields? As the microplastics, migrate through the soil horizons, they are fractioned according to their polymer type, and then fractioned according to the particle size.
That's our hypothesis.
Yes, that's what we were thinking of. That's what was going to be happening.
Yeah, that's our hypothesis.
So then where did our samples actually come from that we researched this summer? They came from, we collected 5 different soil cores from a control field. I keep pointing to the computer, but I'm actually supposed to be pointing up here. So we collected 5 different samples from our control fields, and then from our Biosolid Applied 2022. And from there, what we did was we made a composite of each one of those core samples, and we did a bunch of different studies on those. Go ahead. Okay, so this right here is our, the NOAA method. This is what a lot of kind of the typical everybody had been using. Traditionally, this was oxidizing the organic, to remove the organic matter, and then it was filtered and the organic matter was extracted and separated based on densities And then we got to the quantitative analysis. Okay. However, what we did was we built off of that NOAA method and made a lot of different modifications to 14 modifications in 6 different areas. What I'm going to be showing you today is looking through the digital microscopy and the fluorescent images.
And the density.
And the densities, yes.
Density, sequential density extraction, yeah.
Okay, so here this is showing the fractionation of microplastic particles according to the different densities that we did. We have the residue solids at about a 1.5 grams per milliliter density. Heavy solids and fractions, heavy fractions are about a 1.3 grams per milliliter, and then our light fractions went down to 1.2 grams per milliliter. Okay, so once we have separated, how do we actually quantify? How do we figure out how many microplastics are there really? So with this, we were able to use the digital microscopy, which is the fluorescent imaging. As you can see here on this filter disc, we have our sample, and we were able to look at all the different pieces of microplastics that we're able to see. What we did, how do we actually make it look like this, is we used a dye called Nile Red, which is actually causing those plastics to fluoresce. We can then use imaging software to determine the actual number of microplastics that we're actually seeing in this. And the different sizes, yes. Okay. Here is another example of the Nile Red being used so we can see the different fluorescents. We actually have different colors. As you can see, we have our green, our yellows, and our reds. And as we see the different colors, each one we discovered different types of plastics are these different colors. So you've got your green, which is your polyethylene, polyesters, polyethylene, polyvinyl chloride, yellows, polypropylene, and ethyl vinyl acetate, are reds, polyamides, and polymethyl methacrylate. Yes. Okay. Awesome. Okay. Here is another example of when we used a dye for Nile Red. The top picture is a bright field. We just see under a white light, and then the bottom picture you can see with our fluorescent. What's interesting here is you can see darker fibers, blues, and blacks, but you're actually not going to be seeing them. They actually do not fluoresce, so there's a little bit of a conservative number up there. Excuse me. All right, so based on our data, we figured out that biosolid application increases the light fraction microplastic concentration among all of the horizons. The differentiation between the red, yellow, and green in each soil horizon is being fractioned by polymer type. As you can see, I keep doing it over here, I'm used to being able to move. As you can see here, all the big differences, we actually found almost a nine-fold difference between our 2022 and our control site horizons. Okay. Microplastics particle size distribution was similar for fluorescent particles within our soil horizons. Excuse me. According to the data, we see that our horizons have actually a similar distribution despite the different polymer types. This is an example of the C1 horizons from the 2022 site, as you can see from there. Okay. Also, an important note to note, is just the size distribution is a little skewed. When the particle size gets smaller, the quantity actually gets a little bigger. Okay, awesome. All right. So the particle size within the horizons are similar, and here it is illustrated by the fact that we have a median size values. This is suggesting that microplastics are not fractionated by size, and as they move downward through the soil columns. Interestingly enough. All right, I'll go ahead and re-say for this one. So here we're looking at the different polymer types, but how does that actually apply when we're talking about size? According to our data here, we're actually seeing Microplastic particle size distribution was similar for the fluorescent particles within all the soil horizons. It is a little bit skewed because with increasing particle numbers, it's decreasing in particle diameter, and the median size values is demonstrating the need for detection of smaller than 20 micron sized particles. All right. Median size of microplastics was similar for all of our horizons, which was really interesting. We find out that these particles are not being fractioned by size during the downward transport of these microplastics through our soil columns. Okay. So in conclusions, our biosolid application to soil is increasing the number of light fraction microplastics in all horizons. Different ratios of red, yellow, and green fluorescent particles suggest light fraction microplastic particles are fractioned by polymer type as they are transported downward through our soil column. Similar size distribution of fluorescent light fraction microplastic particles in each horizon show a skewed distribution of increasing particle numbers with decreasing particle diameter. Similar median size values for all horizons are suggesting that light fraction microplastic particles are not separated by size during the downward transportation through the soil column, which is actually what we thought was, we actually thought that was going to be different. We thought that the particles were going to get smaller as they transported down through the soil column. Okay. Acknowledgements for our National Science Foundation, USGS, Dr. Jason Masoner, Dr. Nada Kolpin, and ECU, Dr. Darcy Tessman, and then of course our OU University of Oklahoma, Dr. Mark Nanny, Dr. Joseph Lita, and Dr. Ting Ting Gu.
All right, thank you. Any questions from the audience, virtual and in person?
Start us off if that's okay.
Yeah, go ahead, Peter.
So it looks like we're going from a method where essentially we get a count of a couple hundred microplastics that are larger in size to a method where we get several thousand that are much smaller in size. How is the count for those particles actually performed?
How do we get those numbers, like for the size frequency distribution diagrams?
Yes.
So that's where we use the digital microscopy. Okay. So we stain that. We after we do all the extractions, we filter what we're collecting, and it's on a filter disc. We stain that sample with Nile red fluorescent dye. Then we do fluorescent microscopy. So the polymers are exclusively stained. They exclusively fluoresce. Then, using imaging software, we can count the number of particles and their size and their color. So what's so beautiful about this is an unbiased, no operator bias is in here, okay? And also what's beautiful about it is typical methods for analyzing and quantifying microplastics would take that bright field image and take your infrared spectrometer or your Raman spectrometer and go from particle to particle to particle and analyze it for its polymer type. The problem with that is that you can see from that bright field compared to the fluorescence, a lot of times in the bright field you're going to miss transparent or white or small polymers. Maybe they're even kind of partially hidden by some organic matter, and when you do the fluorescence they just shine. So this is really quite an advancement, and Ting Ting Gu is the master of all things microscopic and imaging software. So she's the one who, I mean, literally, thousands, tens of thousands of data points. So yeah, but it's very exciting because you get this good data.
And horrifying.
Horrifying. Other questions? Yes, sure.
And this is mostly topological, right? If these are going deeper, we won't be able to see them?
You mean the microplastics?
Yeah, so through the specimen, is it mostly surfacial that we are seeing?
No, what we do, so when we collect that core sample, core sample's about maybe a meter or so long. We see an A horizon, and then a C1 horizon and a C2 horizon. So we collected 5 cores in a field, and then we took portions of each one of those horizons from those 5 cores to make a composite. And so what you are seeing here in this data is the analysis of those composites for each one of those fractions. So we're definitely seeing the microplastics moving downward through the soil.
That's
Yeah.
great.
Yeah.
I think there's a slides showing how you're gonna get different horizon, right? There's a
Yeah,
slides.
yeah. Yeah, right there.
Yes,
So,
yeah.
yeah. So this is a fluvial soil, and the A horizon was anywhere from 0 to 1 to 0 to 2 inches thick. Yeah. Other question, Joe.
So perhaps you could comment on the significance of the size fractionation that you've shown, those highly skewed distributions. What's the environmental significance?
All right, could you go to that? So the environmental significance is, so as Amanda noted, as we go to smaller particle diameter sizes, their number increases. But what's really important to realize here is that the median is around 20 microns. The width of a human hair is about about 100 microns thick. So we're talking about something that's a fifth of the width of a hair. And when you get down to 20 microns and below, that is the size that you start having issues with respect to the particles being able to be ingested or inhaled, getting into a bloodstream, and getting transported throughout our bodies. The other aspect is that, trying to find particles that are 20 microns or smaller with standard methodology, infrared, Raman spectroscopy, is really tough. So we are able to get down to 5 microns with the fluorescent microscopy, get these counts. And the environmental impact is that we've got a lot of these really small particles out there. And if you look at the literature, people have been studying. If you remember that paper, that slide that Amanda showed, where they're looking at biosolids, they're looking at, typically microplastics that are 100 microns or larger, or 80 microns or larger. So they're only seeing 100 to maybe a few thousand, while we're seeing tens of thousands of particles that are these small particles. So we've got to kind of really rechange the whole way we're thinking about how to approach this, because it's these small things that really are the public health threat.
Do you have an explanation yet, an idea, a hypothesis for why we don't see a separation in the size of the particles as you go down through the horizon?
I don't know. It's an interesting question, and I've noticed that in some of the other literature that has looked not as extensively as we've done, but there's been some papers out there where they're kind of rough measurements, but they have not seen a dramatic change in size. So I don't know. It's interesting. But I'll say one thing. I think one thing we might be able to say is that maybe these particles are not breaking down into smaller and smaller particles, and once they're in the soil column, that once they get there, they kind of stay there. So, other question? Yeah, go ahead. Out there in the virtual world.
Mark, somewhere real close here, you have a slide showing the proportions of particles in different fraction extraction methods. Yeah, that right there.
Yes.
So, I just wanted to make sure that, what Amanda presented on was just this light fraction, this 5.1%.
Yes, that is correct. So this slide, what you're seeing here is data we collected for the biosolid that was applied to the fields we sampled from. And in this study of this biosolid, we completed the entire comprehensive analysis. So we were able to get the 4 different density fractions, light, heavy, solid, and residual. And the thing that's really, astounding is that the NOAA method would only capture what we call the light fraction. So the early literature using the NOAA method, they're only getting that, that light fraction. When we examined the entire sample, everything, things that would get thrown away in the NOAA method, we find that, we're finding 95% of material plastics in there. Now this is for biosolids, but for the soils that you saw today, we only presented the light fraction data. We have collected the other fractions. They are in process of being stained and analyzed with the digital microscopy. We just did not have that data done yet, but that is in progress.
So Mark, I mean, when you do these other fractions, the results are going to just increase.
Absolutely.
A lot.
Absolutely. Yes. So, now it's, we're talking about, tens of thousands of particles, microplastic particles per gram of dry soil. That's my prediction when we get done with this study. Okay, other questions? Joe?
So I think one of the important points is that the size distribution in the control sites doesn't really dramatically differ from the size distribution in the biosolid amended sites. So that is, you're just putting a lot more of them in the soil, but the distribution seems to be comparable, at least for this fraction.
Yes. So with the control site, obviously we detected some microplastics there. It's not surprising. Air, wind transport rain, etc. But when you looked at that size distribution, those frequency diagrams all look very similar. And I show that in this slide by the fact that the median size value pretty much across the board is, between 40 and 15 microns. So again, this is showing us that, again, we're working with really small particles out there in the soil. The bulk of them are very, very small.
So how much more, in terms of a multiplier, is the amended one versus the control site?
So that's shown. So Joe is asking how many more microplastics do we have in the amended site versus
this fraction.
control? We have about a 9-fold increase in just the light fraction. Okay, well, we're only showing you the light fraction here, but we have 9-fold increase in the number of particles in the biosolid treated soil. And you can see that, for example, like here in C1 of the 2022 soil horizon, we've got quite a bit of this yellow fraction that is absolutely absent from the control site. So there's definite evidence that the microplastics are providing, are being there from the biosolid treatment. All right, other questions out there in the virtual world? Oh, one more here in the audience. Yes, Peter.
I know the focus of this wasn't speciation or anything like that, but I did note that you have the different polymers show up differently with the Nile red dye. Was there a dominant one or anything that we're seeing a lot more than others as far as that goes?
Well, it's interesting. I mean, you can see here, just like on this slide, for the A horizon, we get a predominance of the red. So that is, nylon and methyl acrylate. will show red. Now, that doesn't mean those are the only polymers. Those are the only ones we tested.
Sure.
In the C2, you can see there is a huge yellow presence here. That is polypropylene and ethyl vinyl acetate. Now, again, there could be others, and we're working on it. I mean, it could be polyurethane or something like that. We've got to test these other polymers. In some of the fractions of the biosolid, we would get predominantly green. Half of the particles would be green, and about a quarter would be yellow, and a quarter would be red. So there is this, differentiation among different fractions. What does that exactly mean just yet? I don't know. What I need to do next is now take these samples over to the Raman spectrometer or the microscope and begin to definitively identify these polymers. Tasha? Yeah.
Not a question, but just a comment. Such great work. Work with PMMA, which is shown to seep into cracks as, I'm a concrete person.
Yeah,
I
yeah,
cracks. Seep into as low as 20 micron
Wow.
cracks. My PhD student Ali has worked with some of the material. And it's so interesting here because it could be possible that these are probably getting into the smallest particles.
Yes, yes.
And we need to look into it deeper and deeper because these are harmful materials.
Yes, yeah.
So interesting.
Well, thank you. That's an awesome comment. I'll have to talk more about that.
Yeah, we should talk more now, Mark.
All right. Any? Yeah.
Have you,
Rachel.
so this is just specific to biosolids, but just from a water treatment perspective, have you looked at water treatment residuals at all, or?
I mean, wastewater treatment.
No, just drinking water. 'Cause
Drinking.
Repeat the question.
Yeah. Repeat the question louder for
Yeah.
people in the back.
I was asking if, since this is specific to biosolids, wastewater, since I'm a drinking water person, I was curious about drinking water residuals specifically, since there are polymers added to drinking water in the system when you treat the water, and so I was just curious if you're planning on looking at that in the future or anything, because I know a lot of drinking water facilities are starting to land apply their residuals as well. So.
Yeah. Yeah. No, what Rachel is talking about is something. Yeah, it's a very interesting question. At drinking water treatment plants, they use coagulation and flocculation, sedimentation to remove very, very fine particles. And the idea is, are microplastics being removed in that process, or how many of them are? And that sludge that goes to the bottom then is captured, taken to drying lagoons, and then land applied, because it's mostly like calcium carbonate and iron oxides and things. No, I don't know anybody who has, but that is, I think, a really important question to ask. What is the removal rate? I know people have looked at tap water versus plastic water bottles. Tap water is the way to go if you want to avoid microplastics. But no, if you've got ideas about a project, talk to us. Yes, Jason.
I was just going to jump in there and let Rachel know that I've kind of contacted our water infrastructure IST in conjunction with the food IST, and I have planted a seed for a project related to that deals with sludge and land application of from drinking water, and nothing firm yet, but at least we're talking about it internally at the USGS. Oh, and Mark,
yes,
one more question for you guys. Do you pointed out that as you decrease in particle size, the frequency increases dramatically, yes. Right?
Yes.
So the smaller you get, the more you have. Do you have any ideas about the nano scale? I mean, is this going to continue? And we're seeing, are we going to see 80-90% of microplastic sizes is in the nanoscale?
You know, I wonder that, and it's very tempting to just keep drawing that curve straight up, you know, but that would be, I think, ethically irresponsible as a scare tactic to the general public. But, that's a great question, Jason, and it's one I have wondered about a lot. So I don't know of anybody who's actually tackled that.
I can answer the question. So,
Okay, Ting Ting's got a response.
yeah, it's a very good question. We're also interested. The current technology we're using, the light microscopy, and there's a diffraction limit, the light microscopy. So we could go down to 1 micron, and maybe slightly lower than micron, but because of the diffraction limit, that's the physics, and then we're not breaking the physics, the law.
Yeah.
So we're not able to, with this current method for light microscopy, we can do it down to 1 micron. But if the technology involving, then we actually can using electron microscope to see things smaller than in the nanometer range. But it's going to be not high throughput, right?
Yeah,
So
yeah.
for electron microscopies, then processing the samples and also looking for, it has to be 1 by 1. It's going to be a very slow and time-consuming, label-extensive method. So it's not going to be put into a large data set analysis or high throughput for real-time, data reporting. So with current microscope, we're doing that. We're already breaking the limits for a lot of people in the field. They're only down to like 100 microns or 80 microns, 20. We will be able to go into the micro size.
Yeah.
And that's actually really important because
Yeah.
they can go into the blood vessels, they can go into our
That's right.
vascular system. That's where we should see some correlations, what the health risk and health impact.
Absolutely, yeah. Yeah. So with optical microscopy, our physical limit is at 1 micron, and to go to the nanoscale, we have to turn to another method, and haven't figured out how to make a high throughput method for that yet.
Yeah, with the technology approachings, and
Yeah.
down the road,
All right.
Mark, you're onto something because, the extraction methods, everything can be turned upside down, when you're planning on analyzing for nanos, guys.
Mm-hmm.
You got to rethink everything, really.
Absolutely, yeah.
I mean, I don't know. I was just thinking of like standards. Has anybody ever looked at, how many nanoparticles are in, the microplastic standards you buy and use from NIST,
Yeah, I don't know. Yes, that's a good question. Yeah.
They don't make them.
Joe says they don't make them, which, well, Jason was asking, I guess like a contaminant nanoparticle with the standards we use. Is that what you're asking, Jason?
Yeah,
Yeah.
exactly. We don't know what is coming from contamination at that size range,
Yeah. Well, and then, you're going to have a whole new way of thinking about how you even manage the samples and the solvents and solutions you use to treat the samples during extraction. It's going to require a whole new way of thinking.
Yep, that's what I was getting at.
Yeah. Other questions? Yeah, Ting Ting.
I have a question for the teacher. You guys did a great job with such short
It
time.
was a team effort. It was a big team effort.
Yeah, they worked hard, and Daniel, there's Daniel. Daniel and the teachers, they were just, I stayed out of the lab because they didn't even need it. It was like a restaurant kitchen, they were just running around doing things and kept the lab clean and neat and organized. They were spectacular. Yeah.
What is the one major challenging for you guys thinking about, and then what is the highlights, exciting points from the project? Go on.
Well, I will say sometimes it seemed like there were too many people trying to do too many and sharing too much equipment at the same time, so that got a little challenging. But one of I know for myself, and I don't know if I can speak for everybody, but we were all really excited just being able to be a part of this and seeing the results from everything that we've done. That was the big part for me, especially. I'm just like seeing it come to life. Oh, this is what we found. This is amazing. I didn't know that. And so it was a big learning process.
Yes. I do not have a background in science at all. And I am an elementary school teacher and I teach special ed. So this was completely foreign to me. But my school was built on a former landfill. So when I saw his study on soils, it just sparked an interest. So I have learned so much being here. It's been exciting.
Correct.
Joe, did you have a question?
I guess more of a comment. If we go on the biosolids fractions, that light fraction that you've talked about today is 5% of the issue, right? Maybe 5
Correct.
or 6%. So the other 95% is yet to be uncovered.
That's right.
And so
That's right.
there's a lot of stories there yet.
Yes. Oh, yeah. Yeah. This is just the tip of the iceberg. So,
The more information you find, the more questions you're going to ask too. So.
well, I think what Joe is getting at, the number of particles,
Yeah.
I know that soil is going to, it's going to go up a couple
Right.
orders of magnitude compared to what we see right now. Yep. Yes. Yes.
You know, you guys, and you kind of, you think about, long-term application of biosolids happening year after year after year after year. So it's kind of obvious that the plastics would increase. And
Yes.
one thing that I think about is what we learn, like things like the accumulation of microplastics with continued application on the same farmland. I mean, part of the regulatory management toolbox might be to kind of suggest that we don't use the same plot ground year after year,
Absolutely.
but we rotate it around, once
Yep.
every 3, 4, 5 years,
Yes.
And it's kind of cool to be working on something that has the potential to improve, how things are disposed or regulated in the U.S. under beneficial use.
Absolutely, yeah. I mean, biosolids, the amount we create every day is really unfathomable. And I remember, when I was in grad college in Champaign-Urbana, Illinois, and every day around 4 p.m., this train would come going south from Chicago with all these hopper cars full of Chicago's biosolid waste, taking it somewhere down to southern Illinois. But, and there were like 20, 25 of these hopper cars daily leaving Chicago. So, Joe.
Well, it also gets to the point of how much of that biosolid that was analyzed initially, how reflective of that is?
Yes.
Is that for a month of waste, or is that a year or 6 months? How long before that gets applied to soil?
Yeah, I think the question is what is the heterogeneity of a biosolid sample, and how well can you take a point in time biosolid sample that you analyzed and extrapolate it to what's happening out there in the field?
You said it better than I did.
Yeah.
But looking at the soil, you can kind of deduce.
Yeah,
what's
and work backwards.
into it and the application rate.
Absolutely, you can work backwards, yeah. So, yeah. All right. Yes, Chris.
I'm just wondering, with the application of biosolids, has anyone done experiments where they just stopped doing application of biosolids and then looked at, stopped it on a plot of land and looked at?
Well, actually, that is part of our study. We have, Amanda mentioned we have 3 sites, and the 2015 site we didn't talk about. We don't have we have the filters ready for staining and analysis, but we didn't get to them, analyze them yet. But yeah, that is an example of a site directly addressing what you talked about. So the last application was in 2015. So it had sat there for, 7, 8 years before we sampled it.
And we don't have that data yet.
Well, it's all in my lab sitting. These folks, part of their summer, they process those samples. Those samples have been processed, waiting for digital microscopy.
Wonderful.
Yeah, so they blew my mind how much work they got done this summer.
Many hours was invested.
Yeah.
Blood, sweat, and tears at this point.
Thank you all.
So, Peter.
While we're on the subject of sites, I have a question that might speak to the difficulty of setting up this experiment, and that's soil is incredibly diverse. Site to
Absolutely,
site.
yes.
And so how difficult was it to find 3 sites with matching soil types that would fit this experiment? I bet that wasn't trivial at all.
Well, we were very fortunate this site is reasonably close, and we were able to, it was growing Bermuda grass, and very fortunately, right next door was another farmer who had a field with grass that he had never applied biosolids. And so the soil profiles were identical, and we made sure of that we used USDA soil maps and everything, but then when we were out there in the field, we'd take a soil core and we might we can't use this, it's got too much clay right here, we're gonna go a little bit over this way. And so we worked really hard at that, but yeah, you're absolutely right. These were sandy loams and
Mostly sand.
mostly sand, the
They were
C1 and
down in the
C2.
lower left
Yeah.
corner of it.
Yeah.
That
I
was
was here
managed.
last year as well when we went and pulled the core samples, and we would pull the core sample and examine it and decide to put it back and go find a different spot in the field because we did want the soil to be so similar. So we were out there 2 days straight pulling core samples. So we did a lot of work to make sure that they were very similar.
That was my immediate thought. I was like,
Yeah.
man, they probably pulled a lot of course for this.
Yeah. Well, actually, I was surprised. We did a pretty good job using the soil survey data and then just going out there and, kind of looking around, and you get a feel for the elevation and the slope and kind of use your brain and but yeah, that's a whole other question. Yeah, absolutely, yeah. Other questions? Virtual world? All right.
This is Patty Bright
Yeah.
from USGS. I don't have any questions, but I just wanted to say really nice job on this. And it's, I'm really glad to see you guys doing a topic like this because obviously it is so timely and it's getting a lot of exposure here in DC on the Hill. And certainly an issue that is going to be an important one, I think, for our students to understand. So I'm so glad this is something you're going to be able to take back to your students and talk to them about. And just, this whole issue of plastics is not going away. Honestly, I think every single day I open the Washington Post, there's a new article on plastics and where it's being found in fetal tissue and other things. It's really quite frightening. So thank you so much for the work you guys are doing. This sounds like a really great program.
Oh, you made my day by saying it's frightening. Bet only Stephen King feels that way too. No. But yeah, you're absolutely right. It is really disturbing when you see these size frequency distributions and you realize we're seeing medians of 20, 25 microns, and you think about the bulk of plastics that are smaller than that, and wow, it's...
Well, we often have trouble getting, sometimes these scientific issues are kind of flying under the radar and we have a hard time getting congressional interest in them. This is not one of those, obviously, but, a few years ago we had some intersex fish in the Potomac River, and that's the drinking water for many of our congressmen. So, of course, that really kind of worked up without endocrine disrupting chemicals, which was great. And I was just joking with one of my colleagues last week. There was an article in the Washington Post, and the headline was something like, microplastics found in penises, and I was like, well, good, so that should really get us into
Yes, yeah. I'm not going to say anything more about that.
On that note.
We'll leave it at that, but anyway.
Yeah.
But I just wanted to say, I need to jump to another call, but this was really great. Thank you so much, and thank you for inviting us.
Thank
really
you for
appreciate
attending.
it, and like I said, great program, so.
Well, and we'll keep you up to date as we get more data, and we are in the throes. Actually, I'm really happy because I'm going to double my filtration system. Amanda is going to work in the lab, so full-time, and I've got Daniel hired for the next 2 semesters, so I now have 2 really good trained folks at the bench, and that will speed up our processing. So the LARM samples for the USGS, the deer samples, this study, we will be making a lot of good progress. All right.
That's really great. This is a really great collaboration between the groups. And we're looking really forward to the paper that Hope is coming out soon.
Thank you. Great.
Mhm.